Good evening, everyone, and welcome to our online Academy Discourse this evening, which is supported by the law firm of Mason, Hayes and Curran. I am Mary Canning. I'm President of the Royal Irish Academy, and with us this evening we have the Academy Secretary, Professor Mary O'Dowd, the Secretary for Science, Professor Catherine Godson, and joining us online are our Senior Vice President, Professor Jerry McKenna, and Academy Treasurer, Professor Patrick Honohan, and our Executive Director, Dr. Tony Gaynor. And it's very pleasant to see that this is not just online, because tonight we have one of the largest audiences we've been able to have so far since the beginning of the pandemic. So you're all very welcome. And very welcome to all of those of you who are joining us online. Academy Discourses are the oldest and most renowned series of talks in Ireland. The first discourses were presented in 1786, and traditionally and historically they were an occasion reserved for academics to first reveal and discuss their work and to give a public account and description of what they were doing. We now record these discourses and they are available on the Academy's website. So before we begin, I have a little bit of Academy business to transact, which is always part of each of our discourse evenings. The minutes of the last discourse, which was the future of higher education in Ireland and generally, globally, was a conversation between Professor Sir Adrian Smith and Professor Orla Feely, who's a member of the Academy. And this discourse took place on the 28th of April, 2022, and it is posted online, and we have had very many views of that discourse. Since no member of the Academy has informed us of any issues with the minutes of this discourse, I am proposing to sign them and put them into the minute book at the end of this meeting. So now it is my pleasure now to turn to the business of this evening. Our speaker for this evening's discourse is Professor Lalita Ramakrishnan, and our respondent is Professor Joseph Keane, member of the Royal Irish Academy. Dr. Lalita Ramakrishnan is Professor of Immunology and Infectious Diseases at the University of Cambridge, which she joined after her position at Stanford University and at the University of Washington. She developed there a zebrafish model for tuberculosis, and I have to confess that I don't know what that is, but it's something we're looking forward to learning about. Her laboratory in the University of Cambridge continues to study TB pathogenesis. Professor Ramakrishnan has received many awards and honours, and that they include the NIH Director's Pioneer Award and the Wellcome Trust Principal Research Fellowship. And our respondent tonight is Joseph Keane, member of the Royal Irish Academy. He is Professor of Medicine at Trinity College Dublin and is a consultant respiratory physician at St. James's Hospital. Following the conversation, we will have the opportunity to have some questions from you, our audience here, and also from those of you online. If you wish to ask a question online, please use the Zoom question and answer function, and we hope to have time to answer as many of those as we can. So now to the discourse, and Professor Ramakrishnan, over to you. Thank you very much.
Thank you very much for having me. We'll get started. So I want to tell you a little bit today about a respiratory disease that is not COVID. It's tuberculosis. And while COVID is the new kid on the block, TB is an ancient disease. Depending on who you ask, it's been around for between 30,000 and 70,000 years. But in either case, it predates the Neolithic demographic transition, which means that it's had to adapt to very, very big changes in human behavior, and it still managed to be with us. Now, this is a picture of a mummy from a mere 3,000 years ago. That was, and you can tell from the shape of that spine that the mummy succumbed to TB, because this is a characteristic curvature of the spine called Pott's disease of the spine. Now, this happens to also be my very first introduction to TB, because when I was 6 years old or less, slightly less, my mother got Pott's disease, and she ended up having to be treated. This was in the days when the drugs were much worse than they are today, and she ended up having to take drugs 3 times for a year each time, and ultimately got compassionate use treatment with the most modern drugs, and that's what eventually cured her. So this reflects that this graph very nicely reflects the fact that TB, partly because it's so old, is also humanity's greatest infectious killer. And you know, when you think about global scourges, infectious scourges, the thing that will come to people's minds is the plague or influenza. But look at the contribution of TB versus plague and smallpox, and you'll see that it is vastly greater. And COVID, of course, would just be a little dot, a little speck in the middle of that, of there right now. Now, you know, as I mentioned from my mother's history, the drugs got infinitely better, and so that by the early 70s, we now had what was called short course chemotherapy. But short course chemotherapy just means that instead of treating people for a year with some nasty drugs, you had somewhat better drugs that you could treat them for, and they would get cured in 6 months. But it was 4 drugs for 6 months. And any one of you who's been instructed to take antibiotics for 7 days knows that you can barely do it without skipping a few doses here and there, especially when you start to feel better. And you can imagine how this would be if your livelihood depended on not taking the drug, by not going, if you could skip going to the clinic, and which might be a day's walk away, and if you could instead work, you could imagine that if people start getting better, how they might start deciding not to take treatment with all these many drugs. And so partly as a result of this, TB remains a global scourge. In 2022, it killed 1.5 million people, and possibly more, because it's been difficult to estimate the last year because of COVID, and sickened at least 10 million people. 500,000 of the people of the people who die from TB are children. So it is, it's a terrible, terrible disease. But what's even worse is that now, on top of drug-sensitive TB, we have multi-drug resistant TB. And multi-drug resistant TB can involve extensive drug resistance. And if you lose the modern day drugs that my mother finally got cured from, then you have relegated TB back to the pre-antibiotic era. And we saw a very stark example of this when Joe Keane was kind enough to take me on his TB ward rounds today. And 2 patients who had come in from Ukraine had drug-resistant TB that they had carried with them on the train, on the plane, before getting diagnosed. So this is a very serious extant problem. Now, but the curious thing that we've been thinking about for many years now, which is also brought into relief by the COVID epidemic, is that while TB is such a major scourge and kills way, way, way too many people, actually most people get cured from, if they get infected with TB, they get cured without even knowing that they're infected. So most people self-cure. And this is something that Paul Edelstein, Marcel Baer, and I worked out from doing a very detailed analysis of the epidemiology of TB from all the way from the pre-antibiotic era to now, which we published in a series of papers, which has made us slightly unpopular with the TB community, because which community that studies a disease wants us to undersell the disease? But actually, it's not only interesting, but it also gives you hope, and it gives you a way forward to think about how you might deal with this disease and how you might conquer it. And so most people clear TB, and not only that, they clear it pretty early after infection. And it turns out that most people in this world, contrary to what everybody is saying, including the WHO, do not harbor disease. They do not harbor a latent infection for years and years and years. And so we need not be afraid of people who don't actually have the disease, but rather most people either get it early, and that's a small proportion of the people who get infected, and the rest of them, which is the vast majority of the people who get infected, actually clear the disease altogether. Now, the interesting scientific question from this is, okay, well, what's wrong with the 5 to 10% who don't clear it? What is it about them? And if you of course, it's very tempting to say, oh, they've got a gamma interferon, they've got, deficiencies of known cytokines or known immune protectants. But it turns out that most people who get TB don't have anything obviously wrong with them. They are to all effects immunologically competent, and yet they're getting TB, which means that they have some cryptic immune deficiency, if you want to put it that way, be it specific to TB or not. And this idea, and of course, the most tempting explanation for this is that there are genetic variations in susceptibility to TB, as indeed there are to just about any disease in the world, be they infectious diseases or not infectious diseases. For example, a great example of this would be, say, lung cancer. Joe and I have had many patients who say, oh, I'm not going to give up smoking because, my dad smoked all his life. He didn't get lung cancer. Well, yes, it's true that not everyone who smokes gets lung cancer, but if you have if you add in a genetic susceptibility of some sort, then you will be at higher risk. So same here. So, but the thing that really emphasizes that there's a host genetic susceptibility to TB comes from a tragic experiment that got done accidentally, and this was in the year 1929. Now, one big problem in TB is that we don't have a good vaccine against it. There is a live attenuated vaccine called BCG, and BCG is still used in TB endemic areas just because it protects babies and very young children against a terrible disseminated form of TB. But for mysterious reasons, once they start becoming adults, it doesn't protect at all. And so this is why we still have TB despite the vaccine. Now, BCG was made by just culturing bacteria until they became avirulent, and then back in the day, once they found it was avirulent in animals, they rolled it out in kids. Now, in those days, you couldn't freeze vaccine and send it around. You couldn't freeze bacteria so how did they do it? They sent stabs of the bacteria all around the world, and Joe Keane would be the guy who would grow it up for his hospital, and then he would give it to people. And in this hospital in Lübeck, which is in the north of Germany, why can I never get this to, well, right there, that blue dot, which is in northern Germany, which is genetically supposedly homogeneous. On the 251 kids were given BCG, except the physician had accidentally grown up virulent tuberculosis. He mixed up the vials. So these kids got virulent TB. But look what happened. A third of them died of TB. A third of them absolutely got TB and were sick from it, but they survived. And the remaining third never got sick from it. And the only reason we know they had some TB was when they did x-rays, they could see some signs of it, but otherwise they cleared. So clearly, these kids who got absolutely the same dose, they got a dose the day they were born, and a month later reacted very differently to it. So this is not too surprising if you look at the life cycle of TB, so to speak. So TB is transmitted by bacteria. These bacteria are coughed up by individuals who have the disease. You can see that on the left. And they make their way into the individuals who are nearby to them. And there they can set up, either be cleared or set up shop. The first cell that meets them are these macrophages in the lungs, which are the first defenders in the lung. And many times that interaction can result in the bacterium being killed. But if that doesn't happen, they can actually use the macrophages to get deeper into the interstitial, past the epithelium of the lung, deep into the lung. And there they set up these structures called granulomas. And these granulomas, or tubercles, are a quintessential structure, hallmark structure of TB, where the macrophages band together, they undergo a kind of specialized differentiation, and they also recruit other immune cells. Now the granuloma can end up killing the bugs, but equally the bug can take advantage of the granuloma and use it as their growth niche, and they can replicate within these macrophages. And so all of this depends on the different interactions between the host and the bug, and host genetics can come into each of these. The final stage that is really important is that coming back to the left-hand side again, is where the macrophages die and they release the bacteria into this sort of cheesy debris in the middle called caseum. And it is this necrotic or caseating granuloma that is needed to spread the infection to the next individual. Now remember that there's a big difference between TB and COVID. COVID can transmit without any disease. Disease is an accident. COVID could do just fine if it never killed anybody, because all an infectious agent wants to do is to survive. But TB cannot. The only way TB can survive evolutionarily is to cause disease, and specifically to cause this necrosis, because with the necrosis is how it spreads to the next person and the bug is able to survive. So on this backdrop, I was an infectious diseases fellow, and I really wanted to work on TB. And I decided when I was a postdoc to try. I was encouraged by my postdoc advisor to find a surrogate model that wasn't the human TB bacterium. And what I ended up finding was a bug that is the closest genetic relative of the human TB bacteria. It's called Mycobacterium marinum. And Mycobacterium marinum is well known to infectious diseases physicians, and particularly to dermatologists, because it gives a superficial lesion like this. But the bugs look very similar to TB. And if you were to look at the pathology of this lesion, it would look very much like the granuloma that you see in TB. Now, the reason that, while Mycobacterium marinum is an accidental pathogen of humans, it doesn't spread human to human because it never gets into your lungs but it turns out that it is a natural pathogen of fish. Now, why might that be? Why would, and the fish get deep deep-seated real tuberculosis. In fact, it's called fish tuberculosis. The fish get thin and sick and they die of it. Now, why is it? It's because this pathogen, although it's closely related to TB and is a predecessor and evolutionary ancestor of the human bug, is temperature adapted. It is a natural pathogen of cold-blooded animals. So cold-blooded animals get deep systemic infection. Warm-blooded animals get superficial infection on the surfaces of our body and so on. Now, this is a human granuloma, and you can see how it's nicely and tightly aggregated. And you know, where I've got that arrowhead in the middle is where you can see there's no blue because the nuclei on this H&E hematoxylin eosin stain, the macrophages have died and only the cytoplasm is visible. And if you look at a zebrafish granuloma, which I've in this case stained with a stain that also stains the bacteria, you'll see how closely related it is to the human TB bacterium. It's a cellular aggregate, and you can see where there's areas of cellularity, the bacteria are sparse, and where the cells have died, you can see the bacteria growing like gangbusters because they no longer can be even partially restricted by the macrophages. Okay, and the zebrafish community is well aware that TB is a bad disease of them. This was not our discovery. In fact, they want to have nothing to do with us other than by telephone or email. But the beautiful thing about the zebrafish and why developmental biologists love it is because it has a prolonged larval phase where it is transparent. And it's tiny, it's transparent, you can do live microscopy, you can do all kinds of cool things with it. So we asked ourselves, okay, can we also infect these baby zebrafish, these larvae, and will they get the disease, and will they get a disease that looks anything like adult disease and therefore adult TB? And here is a larva that we've infected, and we've given it, and you can see those collections of bacteria, and they very much look like granulomas. But to remove any doubt, we engineered the fish so that it would have green fluorescent macrophages, the key cell type, and we also had them have red fluorescent neutrophils, which is another cell that is present in smaller numbers in these granulomas. And you can see these nice tight granulomas that have formed in a fish that we've infected with blue fluorescent bacteria, and you can see the occasional neutrophil in there. So this was all really good all those many years ago when we did this. Now, of course, I make the fish look nice and big, but it turns out that the larva is tiny and fits in the cravat of Lincoln. You will notice that I have not used the penny because I don't want to offend the Queen, particularly not at the time of her Platinum Jubilee. And so how do we manipulate these fish? So we had to devise all these methods. You know, when you start a new model, you have to think about how to deal with it. So we can infect the fish in a couple different places. And so here's a fish that we use a little vacuum suction device, hold the fish, and then we've got these hand-pulled glass needles where we've got the bugs in there, and we've got a bit of dye to see what we're doing. And you can see there it goes, and you can see that here, this is our equivalent of the lung. It's the hindbrain ventricle cavity of the fish where macrophages come and look at the bugs. Or you can bypass all that and look directly by injecting the bugs into the vein, and you can do that with this little hockey stick approach, and everything works, and you will get all kinds of nice granulomas when you do it either way. The other innovation is that the other wonderful thing is, do you see how that fish has a great big yolk? Well, it turns out that the fish doesn't need to eat for the first 2 weeks, which is a simple but wonderful thing for us, because you can just put them in those 96 well plates and give them a little bit of seawater-like material, or river water-like material actually, and you can keep them in those plates. You can image them every day, or if you're nervous, you can image them every hour, and you can follow infection like the way it is here with these fluorescent bacteria, and you can quantify it. You can write a little code, and you can quantify infection, and you can see what's going on every single day to your fish. So now we had it all set up, but we come back to our question of why is it that only a few people get TB. And remember, we wanted to do basically the Lubeck experiment. If we mutagenize fish, can we find fish that are more or less susceptible? And at this point, David Tobin entered the lab, who's now a professor at Duke. And David was a neurobiologist from UCSF, and he had actually worked with worms, but he had then gone to Guatemala for a couple of years and gotten very interested in infectious diseases, particularly TB. So he came to my lab and he did a forward genetic screen. And by this I mean we mutagenized the animals and we then got their offspring, and we now examined their level of susceptibility. And you can see above is a wild type sibling, and importantly, these are siblings. And you can see the mutant sibling is way, way more infected than the wild type sibling. And he got several such mutants, but I'm only going to tell you about one of them. So what David realized was that these fish, these mutants had accelerated necrosis of the granuloma, which allowed the bugs to grow wildly. So let me, and what he noticed was that these bugs were growing extracellularly in these serpentine cords that has been a feature known for the TB bacterium ever since Robert Koch identified the bugs in 1881. So let me show you again the granuloma with the cellular the cellular granuloma, and you can see the bugs are just sort of clumping in there a little bit, but they don't have room to form these expansive cords. Now in David's mutant, what you could see was that very quickly the macrophages were dying, and this is the same corresponding time, and you can see there's very little green, and there's a lot of blue because the bugs are starting to grow extracellularly. And what you can see just a couple days later is that the macrophages are almost gone, and the bugs have formed these cords that I'm talking to you about. Okay, so when David mapped the mutant, it mapped to the arachidonic acid pathway, and these are 20-carbon lipids that are present in our cell membranes, and they've been increasingly understood to have all kinds of immunological and signaling properties. And in particular, our mutant mapped to an enzyme called leukotriene A4 hydrolase, or LTA4H, which converts an unstable epoxide intermediate of arachidonic acid to a highly stable pro-inflammatory molecule called leukotriene B4. And what David showed was that if you did not have enough, our fish had insufficient LTA4H. And in that case, they couldn't mount a good inflammation. And because of that, they had uncontrolled bacterial growth. Now, at this point, we were made aware, and we knew and were made aware by our friends that we were working on not even sushi, not even fish, but caviar. And we had better try to figure out if this had anything to do with humans. It turns out that the 1,000 Genome project had just revealed a common variant in the LTA4H promoter of humans, which was functional in the sense, and we proved that it was functional, in the sense that this C to T transition was responsible directly for LTA4H levels. So the CCs made the least LTA4H, and the TTs made the most, and the CTs were in the middle. And we were very fortunate that Guy Thwaites had assembled a TB cohort back then, and we were able to, and he had even saved DNA from it. Well, he had saved blood, which we could make into DNA, and we were able to show that we saw a relationship to TB severity. However, the relationship was a bit of a surprise and a puzzle back then, because we would have predicted that the CCs would do worst, and they did. But look at the other side, look at the TTs. They're doing just as badly as the CCs. And this was a surprise, because why would excess of this nice pro-inflammatory molecule make you feel worse, make you do worse? And so with this, we went back to the fish, and we created engineered a fish that made the equivalent of the human, high LTA4H. And to our great relief, we found that it also made the fish worse. And they also got uncontrolled growth, and they looked exactly like the low LTA4H fish. But what we then figured out was that the responsible culprit was a well-known cytokine called TNF. And Joe Keane, when he was a postdoc in Boston, was the first to show that TNF was a key protective factor in TB, and everyone has reproduced it since, including us in the zebrafish. And so the low LTA4H fish were dying because they didn't make enough TNF. But what we found was that the high LTA4H fish were dying because they made too much TNF. And so this was a Goldilocks model. Too little TNF was bad, and too much TNF was bad. And why was that? So before we went any further, let me just take you back to the human data. So that's our promoter variant. It's 12 bases upstream of the transcriptional start site. This is the data just in normal controls, where we showed that the CCs make the least protein, the TTs make the most. And now let's have a look at the human patient data. The cohort we used was the severest form of TB called TB meningitis. And even with fully drug-sensitive TB, with good treatment, there's anywhere between a 25 and 40% mortality rate. And this is not surprising if you see the brain of this this unfortunate individual who died. You see this gamish at the bottom of the brain. It causes a severe basal meningitis, increased intracranial tension, and these patients die of all kinds of neurological complications. So because of this, people have thought, maybe this has been people have wondered if there was an element of inflammation. And so when corticosteroids, a broadly acting anti-inflammatory drug, came along many decades ago, people started to just use them along with treatment. Many people did, many people didn't. Where I went to medical school in India, we did. But Guy Thwaites decided to do the randomized controlled trial for what was sort of physician-directed treatment or not with adjunctive glucocorticoids or corticosteroids. And when he did the trial, what he noticed was that yes, these people were right. There was a beneficial effect. But look at that effect. It's tiny. This is not a great big effect. But because of this, because any effect is better than no effect, now they became the standard of care. And when we realized what our fish data looked like, where both too little and too much inflammation are equally bad, we realized that maybe the reason for this was a genetic mixed bag, that these patients either had low LTA4H or too much or high LTA4H. And if that was the case, we were in a position to test this. We were in position to test this by going back to their blood, making DNA, and checking to see if the CCs, if only one group, namely the TTs, responded to the glucocorticoids, right? The high inflammation group should be the only group that should do well with the glucocorticoids. And indeed, look at what we saw. It was exactly what we had wondered. So on the left are the placebos, on the right are the dexamethasone. So the highs are doing really badly with the placebo, and look at them with the dexamethasone. In this small cohort, not a single death. The lows are doing maybe a little worse, but this was not a statistically significant difference. So at this point, we started with the fish, which come from the river Ganga in India. And then we went to Vietnam and we did the TB meningitis cohort in collaboration with Guy Thwaites. And then because we had this strange heterozygous effect, which was considered quite odd, we went to a cohort of leprosy patients in Nepal with another bunch of collaborators. And we saw that in leprosy too, those who were CC, that is high inflammation or low inflammation, got the worst kind of leprosy, multibacillary leprosy, and the middle guys got the better kind of leprosy. And this is a good time to acknowledge Guy Thwaites, with whom I've become a long-term collaborator, and many other people who we pulled in to help us with this, mostly clinicians from these countries. We then went on to do more cohorts. And this is, at this point, Laura Whitworth, a human geneticist, joined the lab. And we banded together with a bunch of Cambridge mathematicians, as well as a bunch of clinicians again. And we repeated this trial in Vietnam, a study, they're not trials. And we also did it in Indonesia, so that we now had 2 independent cohorts where, again, we could see that if you gave patients steroids, which is now everybody gets steroids because it's standard of care, that the TTs did the best and the CCs did worse. So this was all looking very good. Now remember, I told you that in the fish, it's all because of TNF. What about in the human? So in the second round, Guy had saved cerebrospinal fluid. And so we could measure TNF. And indeed, we found that survival was associated with lower TNF, which is exactly what you would expect. So TNF was a risk factor. And not only that, but everything so far was to do with this particular genotype LTA4H, which, even though it's common, it's not everybody has got a high LTA4H, even though it's relatively common. And we were able to show that even independent of LTA4H, the high TNF was a risk factor. So high TNF probably coming from other genetic sources, differences in receptor, differences in transcription factors. So we now had established TNF as a host susceptibility factor for TB, even though it's a host resistance factor as well. If you don't have enough, it's no good. If you have it just right, it's good. If you have too much, it's bad. So the discussion had now shifted to TNF. So now the question, we went back to the fish. And by this time, Francisco Roca had joined the lab. He's now got his own lab in Murcia in Spain. And he asked, how is it that too much TNF is bad? So too little TNF, that's a no-brainer. TNF helps macrophages to curtail bacterial growth, and so without it, the bacteria overgrow in the macrophage pops open, and the bugs come out and they die. But the high TNF, when we went backwards and looked in these same fish, we saw that the macrophages actually control everything much better because they've got a lot of TNF on hand. And yet, suddenly, these macrophages that have hardly any bugs in them pop open. Once the bugs come out, there's nothing that the host has to offer, and they start growing a lot, and they look like the other group. So why is high TNF bad? So this is all stuff that Fran has figured out. What Fran realized was that the high TNF was causing infected macrophages to overproduce reactive oxygen species in the mitochondria of that macrophage. And again, this is a nice plug for the fish. You can actually live label the mitochondrial ROS on which you can see on the right there. There's an infected macrophage that's making a lot of ROS. The bugs are blue, the macrophage is green. And then he was able to follow these, and he was able to show that very soon after, the macrophage that gets the ROS expressed dies. So what's going on? And what he was able to show was that, so normally when you make, the way the mitochondria normally make ROS, or at least much of the time, is when electrons flow into the electron transport chain from metabolic processes. They combine with molecular oxygen, and you get a little bit of reactive oxygen species, or ROS, formed during the course of that. And so the electrons are flowing left to right here. And if you use an insecticidal poison called Rotinone, which poisons the complex 1, where the electrons begin their journey, you will increase ROS production. And indeed, when we looked in normal infection, you can see the infection without any high TNF, this is in normal fish, no high TNF, will make a little bit of ROS, which you can see comparing the first black to the first red. And the Rotinone, because it will block the electrons flowing, you'll get more ROS made. And this is what you would expect. And so we had thought, oh, maybe we'll get a little bit more ROS with the high TNF fish, and the Rotinone will give us even more. But in fact, we had the opposite. We had the predicted increase in ROS, but the Rotinone actually made the ROS go away. So that was a feature that was well known to many people, including, I dare say, I'm sure, Luke O'Neill here. And that reflected that there was a curious phenomenon where the electrons, instead of flowing forward in the chain, as good electrons are meant to do, they were flowing backward in the chain, back into complex 1, and the ROS was being produced as a result of that. Now, reverse electron transport occurs when you have an increased pool of coenzyme Q, and in conjunction with a high proton motive force across the inner mitochondrial membrane. And very typically, it's produced when you have an overproduction of succinate that comes from the Krebs cycle. And indeed, it turned out that one of our neighbors in the mitochondrial biology unit had published a paper showing that succinate was the culprit for increased mitochondrial ROS production in the context of heart attacks during the phase when the heart starts when the ischemia goes away, and now the Krebs cycle, which has been stopped up, revs up again, and now you get this accumulation of succinate from this revved up cycle, and that is how you get this reverse electron transport and you get increased ROS. So we wondered if that was what was going on, and we did genetic and pharmacological experiments to show that that was the case, and we then got Mike and Hiran to do some biochemical experiments on these little baby fish, which was no mean feat, but they really know how to do this with their fancy mass spec machines. And they were able to show that indeed, you can see the first column, the first dot is just wild type fish succinate levels, and the second is wild type fish that have high TNF. Now, and you can see that the succinate levels are higher. But we had already worked out the genetics of this pathway and shown that high TNF causes increased ROS by binding to a kinase called RIPK3 and a mitochondrial phosphatase called PGAM5. It doesn't really matter what they are. But our prediction would have been that if you depleted the fish of those genetically, that you should lose the succinate because you lose the ROS, the mROS. And indeed, we saw just that. So to summarize, what we ended up showing was that high TNF causes increased mitochondrial ROS, and that ends up killing the macrophage, and you get this necrotic granuloma, which is bad for the host. And we showed that the reason that TNF caused the high ROS was that when it combines with mycobacteria in an infected cell, it somehow increases uptake of an amino acid called glutamine into the cell. And when glutamine enters the cell, it gets converted to glutamate, which enters the Krebs cycle through forms alpha ketoglutarate, which that revs up the Krebs cycle and causes an excess of succinate, which we had already shown. And this was the mechanism by which we were getting increased ROS. So this was all very nice. However, it left the burning question of, okay, but why does high mitochondrial ROS cause the macrophage to die? What's going on? Why is the macrophage unable to tolerate this? And Fran worked this out as well. Turns out that when TNF binds to its receptor and you get increased RIPK1 and RIPK3 and you make the mitochondrial ROS by the mechanism we just showed you, this mROS is highly diffusible and it goes into the lysosome, which is another organelle of the cell where these bacteria reside. And high mROS, the bugs don't like, it's toxic to them, and that's why we started to see those bugs dying. But before the bugs can be fully killed, the macrophage, the problem is the ROS activates something called acid sphingomyelinase, a lysosomal enzyme. And acid sphingomyelinase makes, its product is a lipid called ceramide. And ceramide activates a protease, an enzyme called cathepsin D. Cathepsin D makes its way into the cytosol and activates a cell death pathway. And specifically, it activates a protein called BID, and BID activates a canonical cell death protein called BAX. Now things got very interesting here, because BAX is a well-known cell death protein. And the way BAX works is to go to the outer membrane of the mitochondrion, and it forms a pore in there and causes loss of cytochrome C, and that's how the cell dies. But what we had already shown was that this mode of death was not dependent on cytochrome C. It wasn't what is called apoptosis, which is the classic cytochrome C dependent death. Rather, it was some other form of death. So we knew that. And on top of that, in the fish, we could genetically target the BAX to go either to the mitochondrion or not. And when we targeted the BAX to go strictly to the mitochondrion, the cell didn't die. Meaning the BAX wasn't working in the mitochondrion. And we found that instead it was working in the ER, which is another organelle of the cell, which among many other things happens to be the calcium repository of the cell. So when the cell takes up calcium, the calcium goes and sits there and then distributes to other organelles in the cell. And what we showed was that BAX activates a receptor that transmits calcium out from the ER to the mitochondrion. And that we showed that through a channel formed between the ryanodine receptor and the mitochondrial calcium uniporter, or MCU, the calcium flows into the mitochondrion, and when there's excess calcium in the mitochondrion, it signals a cell death protein called cyclophilin D, or CYPD, to go to the inner membrane of the mitochondrion and activate a pore called the mitochondrial transition pore. And when that happens, the cell loses voltage potential and it dies. So this was the pathway that Fran figured out. So we now could show that TNF induces mitochondrial ROS, which elaborates this necrosis pathway involving many organelles, this sort of almost fantastical pathway. So that's all about TNF. But I want to take you back to the idea that TB's been with us for 70,000 years, and it has. And the key thing we noticed was that if you had a high TNF host, not all the macrophages die. Only the infected ones die. Even the neighboring macrophage doesn't die, and that's because the bug is needed along with the high TNF. So there's the genetic perturbation that causes high TNF that's required, but some feature of the bug is required. We don't know what it is yet, but we can tell you it's live bugs that are needed to activate the mitochondrial ROS. And even after the mitochondrial ROS is activated, the bug is needed in a second step to activate the necrosis. And of course, these are very active areas that we are studying right now. So another way to think about all this is that mycobacteria have figured out how to take advantage of host genetic vulnerabilities to cause disease because they need it. Fine, I'm going to end by telling you that, so that's all very nice, but I've given you a very elaborate pathway, which I'm sure you've completely forgotten by now. And so what? Well, it turns out that this pathway gives you room for interventions. And I will begin with pointing out that there's a drug called desipramine, which is a tricyclic antidepressant, but it turns out that it inhibits acid sphingomyelinase. And we were able to show that if you treat the fish with desipramine, you lose susceptibility by reducing macrophage death. Similarly, you can inhibit the ryanodine receptor with another well-known drug called dantrolene, and once again, you lose susceptibility. Fran then had a really clever idea. Some really well-known drugs, pennies to a pill, are what are called calcium channel inhibitors, and we use them for high blood pressure and all kinds of things. And they're very safe, They've been around for a long time. And they inhibit calcium channels, and they will just slightly reduce the calcium burden of a cell. And Fran figured that would also reduce the burden of calcium in the ER just enough. And when he tested that, sure enough, the susceptibility went away. And then finally, coming back to that first part of the pathway where I showed you that TNF induces mitochondrial ROS in the first place to trigger this pathway, there happens to be a well-known anti-diabetic drug, and one of its effects is that it inhibits complex 1 of that electron transport chain. And when we use metformin, this drug that is used anyway in much of the world, again, the susceptibility went away. So to summarize, I've shown you a couple of different pathways, and what I hope I've brought home to you is that mycobacteria, by virtue of being around for so long and being selected, evolutionarily selected, turn out to be exquisite cell biologists, biochemists, immunologists, microbiologists. And because I work in a basic science institution, for sort of half of my position is at the LMB, we have to always remind them that by studying a disease and by studying an infection, you can uncover new biology. So for example, we've brought up a new condition in which reverse electron transport is important. We've shown that cathepsin D, which is a lysosomal protein, is doing all its business outside of the lysosome in the cytosol. What the heck is going on there and how? We've shown that, and most interesting, we've uncovered a completely new mode of cell death, with people studying cell death for 30 years haven't come up with, not because they're very, very smart people, but they haven't used a bug. We've had the benefit of the smartness of the bug. And a wonderful final example is, who knew that BACs would interact with the ryanodine receptor to cause death? And how do we, what's going on there? A completely new function for a 30, 40-year-old molecule. For physicians, such as myself, what we've identified is new drugs, as I pointed out to you. And the beauty of these drugs is that, remember, because they target the host, they're important. They could be potentially used for drug-sensitive and drug-resistant TB, because the drug-resistant bug is going to have the same pathway. And nowadays, everyone's talking about precision medicine, patient-guided therapies, genotype-guided therapies. This is an ideal example of precision medicine, where people with high TNF benefit from this particular thing. But the plug for following the pathway, going deep into the pathway, is that steroids are, they're a sledgehammer, and they have many side effects. And if you have low inflammation, you do even worse with the steroids. But if you use these drugs, they'll be beneficial to the people who have the pathway operant, but they will be neutral to people who don't, because you'll just get them and they'll be fine. And my final lesson for myself is that TB has always been thought to be a disease of failed inflammation. And the old-fashioned thinking used to be that inflammation evolved to protect against infection, which was generally true, and that in the modern era, inflammations come to bite us with heart attacks and arthritis and all, brain diseases and so on. But obviously, life is more complicated. Infectious diseases can also have dysregulated inflammation. And one question we've thought about is whether TNF, whether some of this pathway in some form or the other could be operant in other inflammatory diseases. And I'm going to end with a even broader lesson. So if you have forgotten absolutely everything, then maybe you will remember this. So we're taking, this is not the Ganges River. This is not where the fish comes from. This is a river in the United States. It's on the Montana-Idaho border, and or is it maybe it's the Montana-Wyoming border, but in any case, it is in Yellowstone National Park, and you weren't allowed to swim in this, bathe in this river, but everybody did, and they finally just gave up on it, and they made it so that you had to walk about half a kilometer down the river and get in there, because everyone would, people would be hot from hiking and they'd go in there. And we're walking along, and my husband, who's in the audience, who's the poor guy has had to deal with these stories from me for, every day, on a daily basis, he says to me, "Hey, look, this is Goldilocks." And why might that be? What are you noticing about this? Where are these people in the river? Are they in a straight line? This is kind of how we had to walk out to recess when I was at the Convent of Jesus and Mary all of my life. Yes. And then you get into the river and you can see it even more clearly. Everybody's right in the middle. And why is that? It's because this is a cold mountain river fed by a hot spring. So on the left-hand side, you've got it's boiling. You can't possibly go in there. And if you go even a little bit in the other direction, you will freeze. And so the only place that's comfortable is in the middle. And I would argue that you do best with TB, and you can be in that 90 to 95% if you have the right level of inflammation. But I will also say that if you don't, we're slowly starting to work out ways to get you back in the middle with our host targeting treatments. And I will end with a plug for our fish and stop there. Okay, now I'm terrified because now the discourse begins. Yes.
Thank you so much, Lally. Thank you so much. That was magnificent, fantastic.
Mm-hmm. And what am I supposed to do now?
You're now supposed to retire for a moment whilst I take. Yes, that'll do.
Okay.
No, that was disruptive.
Oh dear.
Yes. So you are one of the most disruptive scientists in the field, and that's why Keystone symposiums are rarely organized without you speaking, because the RAI and TB research needs disruptive people. In the 20th century, we had a very disruptive tuberculosis person called Dorothy Stopford Price. And every now and then, Donald Cox, who is one of our senior scientists, organizes a seminar on a Thursday morning, and he uses the Dorothy Stopford Price room. And she was an advocate for TB, and she fought the hierarchy. And this was in the pre-antibiotic era. And she couldn't be bullied. And she was absolutely right. And I bring her up today because she was there at the time when we had no drugs for this wretched disease. The only thing she had was the BCG, and she tried to promulgate that. I'm not saying John Charles McQuaid helped her, but President, there is no John Charles McQuaid seminar room in St. James's. A little earlier, you kindly did a ward round. This is our patient here, right upper lobe infection, multiple drug-resistant TB, comes from the Ukraine, he gets a text. You go and see somebody, actually the TB you have is multiple drug-resistant. This other Ukrainian gentleman we just discharged, So this is the availability heuristic. If people actually see it right now in St. James's, they think maybe this is important. I think this is important. I think at St. James's, we need a 16-bedded unit to look after these patients, and we need Laura Gleason to come back and look after them in a couple of weeks. In St. James's, we also have a bronchoscopy room. That means that we have access to a remarkable option, which is studying macrophages, alveolar macrophages, and this is why Lally comes and collaborates with us sometimes. And the consequence is that patients who come to St. James's allow us to use some extra cells just for leukoneozidification. These are totally normal nurses. It's just our bronchoscopist is very tall. So Lally has an outstanding model, which is the fish model. It is remarkable, and this is the element I want you to reflect on. It's spectacular. It's not just one cell, which is what we often look at. And it's a dynamic process, and she can track it. And as a consequence, she's made some terrific breakthroughs. Often her fish are quite young, and they may not have much in the line of T cells, and they too are important, as I'm sure Pat Walsh will attest. And as well as that, they may lack other interactions which require looking at, including epithelial cells, which we discussed this morning with Mary O'Sullivan's student. This poor kleptomaniac has been getting a hard time today. In the past, we've shown that MAL, which is a rather interesting gene, behaves in a very similar manner to TNF when it comes to TB. Too hot is no good, too cold is no good, and the right amount of MAL is on the money. TNF has been described as a Janus-faced cytokine. In certain circumstances, it's good, as you mentioned, and in certain circumstances, it's bad. And that's one of the key elements we've got to take home today. Now, in St. James's, we routinely are concerned about patients who take TNF blockers, that they might end up with TB, they might end up with disseminated TB. So we don't use TNF blockers lightly. We're very cautious. However, this patient here had a dreadful paradoxical reaction and a very sore back because her vertebrae were all inflamed after TB, years after TB. And for the first time since I wrote an article suggesting that you should be cautious using TNF blockers, I actually gave it to a patient. Within 1 week, she was better, proving that in certain types of TB, you need TNF blockers. This gentleman was sitting in his kitchen when he fell down. He fell down because he had a paradoxical reaction in his brain. He had a paradoxical reaction in his brain because even though he was on TB treatment, he was taken off TNF blockers, and this paradoxical reaction occurs. Conor Grant wants to know, should we leave these patients on TNF blockers? They don't need the TNF if you've got effective chemotherapy. And Lally certainly has addressed the notion of TNF being dangerous, but also being very helpful in TB. Now, I'm supposed to make a comment that's contradictory to you, Lally, so I hope you feel a bit uncomfortable with these next comments.
Terrible.
But, you know, the leukotriene story is very, very compelling. Certainly the TNF story now and the CSF is also very compelling. But it's a very complex process, the old immune system, isn't it? And is it really going to be down to one biomarker, or is it going to be down to a bunch? So our dear old friend Bonferroni means if you look at enough different indices, you're bound to come up with a pattern, which of course doesn't really help. But Andrew DiNardo has looked at patterns. He's looked at the available data sets on TB patients, and he's come up with a 50-gene classifier. Well, of course he did. I mean, that's a mathematical, that's evident. It's going to happen. But what he suggests is that we can divide patients endotypically, not phenotypically, endotypically, into two groups. And the difference is immunometabolism. So we have the hyporesponsive group who are immunometabolically exhausted, which Lord Leeson is a term that Lord Leeson coined. These immunometabolic people who are exhausted, they're not going to benefit from steroids. But the ones who are immunoresponsive and their glycolysis is going nuts, they're going to benefit from steroids. And he's taken the available host-directed therapies and he's matched them up with these endotypes, which is a good start. But that's all it is. It's a new idea that we're looking at the wrong thing by just looking at one gene. We're looking at the wrong thing just looking at the chest X-ray and the sputum. We need to dig deep. We need to get endotypes. And I think that's something to reflect on. It's blue skies, and therefore SFI wouldn't fund it. But, sorry, Bernie, I didn't mean to upset you there. But that's something where we need to look. And there are new treatments coming that we can certainly fold into that. Mary O'Sullivan has developed all-trans-retinoic acid as a new treatment. We've mentioned that. It works in the mouse, work that she did with Brian Robinson in London. Those are my comments. That's all I would like to say. I look forward to the questions and answers. And I've come in on time, I bet, have I? Good. Thank you. So I'm going to invite people to sit up. Yes.
Yeah. Okay.
Relocate. Sorry,
Okay, well,
Catherine.
Well, thank you both very much for absolutely brilliant presentations. I think everybody will agree they were amazing. Can I just ask, Lally, this is like a kind of obvious question, but if we look at the different genotype responses to DEX in the TB patients, what can that tell us about the heterogeneity of responses to DEX in COVID? And by extension, instead of tocilizumab, should it not have been Humira that was being looked at?
I would argue that it tells you almost nothing. about because, I think we have to be very, I think the reason is that we've uncovered a very specific set of pathways, and I think that's why I really wanted to emphasize how the bug is involved. This is causing disease by killing the macrophage and basically letting the bugs out of the zoo, right? I mean, these are escaped, they're out of the cage. That's how you can think about it. And I think if we, it's very tempting to say, oh yeah, this is COVID is the same. I think the general principle that increased inflammation is bad is going to apply to most infectious diseases. The specifics are not necessarily going to apply to other infectious diseases. But I think it's more a learning thing to say, we've got to think about it. And it's also a lesson in an approach. Like, can you dig in and can you find out what's going on? In fact, I would be more tempted to wonder if conditions that are not infectious but involve a lot of cell death, cell necrosis, whether those might be more related, which involve both TNF and a lot of necrosis. You could imagine rheumatoid arthritis, Crohn's disease, because what you could imagine is maybe there's some other trigger that is not the bug that can substitute for the bug, so to speak, and allow this pathway to come through. So I think that these are worthwhile questions to think about. I'd be very careful not to overclaim beyond the specifics here.
And what do you think the role of the macrophage in clearing up the phagocytic process of clearing up the damage from the dead cells is? And
that is a bit of a mystery, because one question that comes to mind is, why is it that new macrophages can't come and eat these bugs? And there's some data in the fish from other groups, actually, not so much, a little bit from us, but better data from other groups. It somehow seems as if these bugs that become corded because they've had time to hang out outside seem to be less susceptible to phagocytosis from new macrophages than the newly infecting bug. Because if we put in bugs into a fish, even if you put in 50 bugs, they get eaten up. And yet somehow when they come out of this macrophage, it's that corded behavior. And this is another area that we think is something that is really worth investigating, because not only will it be interesting in the disease pathology, but it will also tell you about macrophage biology, like what makes something unpalatable to a macrophage and why.
So for macrophage question specifically, so David McHugh, one of my colleagues in UCD, who said a fantastic talk, he mentioned that he's working with Professor Steve Gordon, who's here, and they're working in macrophage TB infection in cattle, and he was wondering about the epigenetic regulation of the macrophages in humans and zebrafish. Would that be a likely question, Steve?
That could be I don't quite understand the question. Can you explain the question to me?
Not really. It's more about the epigenome, that you've seen changes, epigenetic changes.
Hi. Yeah, I suppose it's about how.
If you see them in fish or in.
Yeah, whether I suppose I can't speak for Dave. So, I suppose what we're looking at here is, in our own work, is seeing how the epigenome is varying in response to infection. And then in terms of how that's varying, we're looking at different pathogens. So for example, infecting with M. tuberculosis into a bovine system versus M. tuberculosis. So I suppose it's that variation that you've eloquently spoken about, and how the epigenome may also layer on top of that.
Yes, so remember that, I've told you that you get high TNF from source. In the human data clearly tell us that you get high TNF, that's pathological, from a fixed LTA4H high genotype, but also from many other sources that we haven't yet identified. I dare say that some of those may be epigenetic. I don't know. They could be genetic or they could be epigenetic. The LTA4H is absolutely genetic. Whether there's an epigenetic overlay, maybe. Maybe some of those who don't have the genotype will have some epigenetic reason, so to speak, of having to have more, express more of it. So yes, you could have epigenetic changes as well. Definitely. I mean, that's a very fashionable area, and I dare say and just because it's fashionable doesn't mean it's wrong. It might even be correct.
I can't. I'm a bit blinded by the light, so I can't.
Me
see.
too.
Yeah, Luke, you have a question.
Lovely stuff. I've got about two other metabolites, itaconate and fumarate, right? So let me start with fumarate. We've got some recent data, amazingly, in macrophages. Fumarate accumulates as well, and it drives TNF production. It drives type 1 interferon production, and it suppresses IL-10. So I'm wondering about fumarate in the context of TB now. Have you ever measured it or seen it in your metabolomic analyses? Or
Yeah, we have. That's a very interesting question, and we ought to. So we specifically only measured because we did the whole thing genetically. We chased after what we saw, and so we looked at succinate, but we could equally now look at fumarate and see if it's contributing. Now, in the fish, for this specific thing, TNF alone seems to be the culprit, because if in the fish, what you can do, which is really cool, is you can make a high LTA4H fish, and now you can titrate down the TNF, and you can show that now susceptibility completely goes away. But that's that's looking at a very specific stage of the infection and of the disease and a particular derangement. Whether fumarate will play a role, I would be very surprised if it did not.
Well, what's great is fumarate can be turned into succinate as well, remember. So I think that's a really good one to measure.
Exactly.
And fumarate hydratase is down, for example,
Yeah.
in interferonopathies.
Yeah.
So I'm wondering, maybe in TB patients there's less FH, and that allows fumarate to accumulate. The other quick one is, has dimethyl fumarate ever been considered as a therapy? Because obviously it's used in MS, as you know, and it'd be very interesting to try DMF, I think, to be honest, maybe in the zebrafish as well.
Yeah. We could definitely try it, because we have tried, you can put it, part of how we figured out all this was by using things like diethyl succinate and so on. You could give these cell permeant succinates. So I'm sure that fumarate will, I'm sure we could do that experiment, and why don't we try it?
Great. And very lastly, sorry, itaconate. There's great literature on RG1 knockouts being really severely affected, and itaconate can block SDH. And maybe that's a way to allow succinate accumulation to drive a separate pathway, perhaps.
Yes.
Did you ever look at that in the zebrafish or RG1?
IRG1 we've started to look at, and we have RG1 fish are, as I'd have to go back and look, but I'm pretty sure they're susceptible, hyper susceptible. But I don't record that bit. And if you're listening, ignore it. But yes. But I think I'm right about that one.
Can I just ask you one thing while the next speaker, the next question is coming? Do fish have all of the TNF mammalian equivalent components? Like, do they have STNF? Do they have the soluble receptors?
Yep.
Can they?
Yep.
Okay.
Yeah, that they're very well behaved. Okay. And I think what it tells you is how basal and important these are. They also, by the way, have gamma, they have type 1s. The type 1s are a little less conserved. They have them, but they're less conserved. And I suspect they're doing the same thing, but it makes it harder to work with them. But gamma is the same, it's conserved the same. And TNF is extremely well conserved, down to TNF receptor 1, receptor 2.
Okay, that's...
all of this. But, just to follow up on Luke's question, we've sort of come hurtling into immunometabolism as sort of, as Mary Claire calls it, if you take a genetics approach, geneticists celebrate ignorance, we're suddenly thrust into this. And now, of course, then what happens is then you start getting interested, because now you start seeing other things. And I think we're very much in that phase right now, where we are looking at, are there related pathways and derangements? And completely separately from this, we've also figured out through a completely different mutant, also from the same screen, that if you tamp down mitochondrial metabolism too much, and in this case it was by using an mTOR mutant, now the animals, again, and human cells do really badly. And they do really badly because they can't withstand a specific bacterial determinant, and we even know what that bacterial determinant is. And so it's a cytotoxin that in this case is functioning as a mitotoxin. And if you can if you can rev up mitochondrial metabolism enough, then you can withstand it. But again, it's kind of a Goldilocks thing, because if you've got to keep your Krebs cycle running enough, or you will die from TB because the cells will go kaput because they simply cannot withstand. They get mitochondrial damage and die. But on the other hand, if you rev it up too much, as you do by feeding in this particular case too much, alpha Kg into the pathway, then you die because now you push your electrons in reverse. So it is this sort of it's really that image in the end that I like so much, that you just have to be right in the middle.
That was such a good talk. Thank you so much. I was just wondering, kind of related to what you were just saying there, when you use the different inhibitors that like to target the pathway you had uncovered, so metformin and calcium channel blockers and things like that, did you try using them in your LTA4 low model at all?
Yes, we did. And thank you for that question. That's what gives us confidence that they won't hurt the reverse. So if you use, we tested high, normal, and low, and everything behaves as you would predict. In other words, they don't affect the low. They don't make the low worse.
But the low are still bad.
Yeah.
But they're not worse.
The low are bad, but they're not worse.
Okay.
They're neither better nor worse.
Which is unlike the steroids.
Correct. And the steroids in the fish make the fish much worse.
In the low.
Yeah, they make the low fish worse. In humans, we don't have enough data to say that they make them worse, but we just don't have enough statistical
We do have
data.
in COVID.
No,
in
in
that
COVID,
trial,
yeah.
In that trial, if you
Yeah.
weren't sick with COVID and you got
Yeah.
steroids, you did worse.
You did way worse, yeah.
Thank you. That was a fantastic talk. I like the idea of repurposing old-fashioned drugs and treating infected zebrafish with antidepressants. It must be like all their Christmases at once. I'm a pediatric infectious diseases physician, and I just want to take you back to, spent many a long hour in TB clinics, mainly treating exposed but infected but not diseased children to prevent, to treat latent TB. So have we been wasting our time? You kind of implied that
No.
they were always called the canary in the coal mine, which I imagine is an analogy that we might phase out over the next 5 years.
Okay,
but,
so this is now I'm putting on my epidemiology hat, and we have no quarrel with the current practices anywhere, neither here nor in any other country. In fact, we've carefully studied the guidelines. You're doing the right thing. But remember, most kids, if you have skin test positive kids, in high burden countries, unless they're recent converts, you're not gonna treat them. And so, but in a place where you have very little TB, you want to treat them for that small risk, and also keep in mind that, and this we published this. If you look at our 2018 BMJ paper, if you look at the data from Amsterdam, kids have a 50% chance. They have a much higher chance of getting TB from an infection. The adults have a 5% chance, And you could see that in old studies and you could see it in modern studies. But kids have a much higher risk, which is why even in high burden countries where it's so difficult, if you find a kid, if you find a kid whose mother has TB, you're going to treat them even if it's a high burden place. So you're doing absolutely the right thing. But, look, we're bringing these things up not to change your practice or my practice where I practice, but to bring awareness to what the actual issues in TB are, and really to say that the vast burden of TB in this world is not coming from people who are activating their latent TB. That's a very small minority, because most people actually just clear their TB. It's a small proportion that will keep going and have latent TB. And even if we, even in the non-pediatric world, we will see that kind of TB here along with immigrant TB. We'll see the old people in the nursing homes who got TB many decades ago. But that's a minority. And to think and to use that as it is a confirmation bias, and it is not what's driving the TB, global TB burden.
Hi, lovely talk. I just have one question. Of the 5 to 10% of people who are susceptible, how much of those do you know are accounted for by the genotype and dysregulation of TNF?
I'm going to get a completely hand-wavy answer. The genotype, so the genotype's very interesting. It is a common genotype, and by that I mean that it is, the allele frequency is about maybe anywhere in the 30 to 40% widely distributed in Asia, wherever you look. But so that means that only about 10 to 15% of the patients will have the homozygote genotype. The genotype is relatively rare in Europe, and it's in between in Africa, which is very unusual for any genetic variation. Usually Africa is at one end. So that in itself is quite odd. And so I would say the genotype, it'll depend on where you are, but it'll account for a much smaller amount. However, Joe and I have discussed this before. Steroids people have always thought steroids had a bit of a benefit, no matter which the population. Veteran studies in the US, studies in the UK. So even among European populations, they've had a little bit of an effect that has never been quite enough. And remember that's why we were delighted to see that TNF was an independent variable, even independent of the LTA4H. So that was very nice. So I don't know how, but that's where I get completely hand-wavy. I don't know what proportion will be because of this pathway and TNF. And again, I would not want to overclaim. I would say this is an important pathway. I would say that it is both important and we should look for it, and we should try to formulate studies and trials to treat accordingly. But this also exemplifies how we should go about doing it and look for other pathways. We're still looking at new mutants because we are hoping to uncover other pathways.
Thank you. Just one, this will have to be the last question.
Oh dear, because I have one comment and two questions. So the first is, I think it's really interesting to think about the evolutionary pressure of tuberculosis. It's been such a widespread driver of human evolution, so it's absolutely fascinating. My two questions are, firstly, do you think that making the diagnosis of TB early enough and intervening early enough with immunomodulatory treatments may be a challenge? Because often, as I'm an infectious diseases clinician, often by the time people present, they have very advanced disease, and it may be harder to modulate that immune response. In your fish, do you pre-treat with the disease-modifying agents before you give the infection, or is the infection already underway before you go in with your calcium channel blockers and so on?
We've done it both ways. Mm-hmm. So, but I would never want to claim that directly translates to humans, because our whole window of time is pretty short. And we have to only think of these things as proof of, as a, not even proof, kind of a proof of principle, a guide, an exciting avenue to think about. But you're absolutely right, because you could argue it's way too late by the time we've got a patient whose macrophages are already caseating and there's inflammation brewing and bugs are spilling all over the place. And that's why, in a way, but remember, that's why it was so surprising. I mean, we were astounded when we saw the human data. I remember I was at a Keystone meeting, and David Tobin, who was still a postdoc in my lab, I was sitting with Joel Ernst having a glass of wine or something, and David came down to me because he was going to give a talk, and they had just finished the genotyping, and he came down to me and he said, he was a very polite man, and he said, "Can I talk to you for a minute? It's important." And he said, "Look at this," you know? And I mean, because of the very question you're asking, it is shocking.
It is absolutely.
It is absolutely shocking how good it is. I mean, you know,
with the zebrafish data,
right.
and then you have actually.
But you know, as my mother used to say, there's many a slip twixt the cup and the lip, and so I want to be very careful. You know, yes, in TB meningitis in Vietnam and Indonesia, and in leprosy in Nepal, we see this. You know, trials need to be done, and I believe they're starting to be done, or studies, to see whether this has any impact in pulmonary TB. I think it will, because, Joe, again, people have been seeing that steroids might be beneficial in pulmonary TB. But if you know, the thing is, if you drown out the relevant genotypes in a slew of non-relevance, you're going to miss effects. You know, and so I think it's worth looking at. But absolutely, immunomodulatory therapies are going to be way more important if you can get early diagnosis, and if you can get early diagnosis
in very resource.
And that is the holy grail. I mean, gosh, if we could get early diagnosis, it would be wonderful. And I'll close by saying that is part of the good news. This is why we're happy with finding that only 5% get TB, because it redirects research from the impossibility of trying to treat 2 billion people with drugs that at some level are hepatotoxic, to saying, guys, why don't we try to figure out which of those people, can we get biomarkers? Can we understand the biology and identify which people are going to go on to TB, which 5%? And this should be what we should be working on.
Which I think rounds back to Joe's proposal of the 50 endotypes, right?
There you go. There you go. Thank you. Yes, absolutely.
Okay.
No, with respect to the steroids, Donald Cox's student, Lorraine Fong, shows you put steroids in a macrophage, you turn off Warburg, you turn off cytokines, and you improve the ability of the macrophage to kill TB. So
but not if you don't have drugs along.
I think of course. But I think there's more to it than that.
Yeah.
I loved your comment earlier about resolution. TB is a failure of resolution.
Yeah.
People aren't studying that. You're not studying that. I'm not studying that.
There's a Keystone meeting on it in 2 weeks.
But that's a good thing to look at. Lipoxygens will be important, but so too will erythrocytosis. So they've looked at...
Mediated by lipoxygens?
They absolutely, and they've looked specifically at the eat-me signal in carotid PET positive, Warburg positive lesions. If you turn on erythrocytosis, the Warburg goes away, the PET goes off, you improve inflammation. So
you change the macrophage phenotype as well.
That's right. We need to look at that.
Can I thank everybody very much, for our speakers and the audience here and at home for fantastic contributions.
Thank you.
So, just a couple of comments at the end. As you all know, the Royal Irish Academy is not just a science, it's also science, and it's the polite literature and antiquities, i.e. humanities. So, listening to this tonight, I was struck by a couple of things, and I'm going to be very brief. When Lalita put up that slide, I thought, yes, TB, such an enormous disease. And of course, as a literature person, there we see it in the literature right throughout the ages. And that sort of came across to me as a completely outsider from this world. And I think you will all agree with me, that was totally fascinating. The world of fish, Goldilocks, I could think about many fairy tale allusions.
Very literary.
And yes, so I think, Lalita, if I may call you, and Joe, I think it's a fascinating discussion tonight. you have brought it to a level where even a layperson who doesn't understand can see the patterns and can understand that this is what the Royal Irish Academy does, which is bring people into a room and let them explain and listen to the wonderful research you're doing. So I think you'll all agree with me, that was wonderful. I'd like to thank our sponsors, Mason, Hayes and Curran, who make this series of discourses possible. The next one is in University College Cork, and it is going to be on decoding the past to conserve our future. And that will be by Dr. Larissa De Santis of Vanderbilt University, and it will take place in University College Cork online as well on Monday, the 18th of July at 8 p.m, and all the details are on the Academy website. So, to those of you who are here, will you please join me in thanking our really inspirational guest and you, Joe, from down the road. Thank you very much.